other note, I can't find any trace of it on the website, but I've seen signs around March 3rd, which is next Monday, is the Health Sciences Career Fair. That nobody seems to know about it. If it happens, there's an advantage for you guys. So you may want to come to that. I was going to encourage you to look at the signs. They're out here in the Fun and Science. I would encourage you to have a resume ready in case somebody wants it when you show up there. Looking like showing up and having your act together is a good part of getting the job. So I'll try to post something on D2L when I see an official notice of that, but that should be coming up next week. All right, 13, reoviruses. Named so because they infect within the respiratory, enteric, tracts, along with some orphans, so things that don't fit into the pattern of respiratory and enteric tracts. They are double-stranded RNA, icosahedral, and naked. Do note we are seeing a variety of genomes. We've mostly seen icosahedral so far, and whether they're enveloped or not. So they are promiscuous. We find reoviruses infecting all domains of life. We're most concerned about them as people because of their respiratory and enteric viruses. The prime candidate is rotavirus. Named so because it looks like a wheel, Latin from rota for wheel. So here's the schematic. And then there's a cutaway based upon electron cryo microscopy. This is a major cause of gastroenteritis. Leading to diarrhea, which of course is not that serious a problem here in the U.S. because hydration with good water is not an issue. But the sad part here, 500,000 child deaths a year in the developing world. So as a virologist, as budding virologists, you'll learn that the thing about viruses is there are not very many very effective treatments for it. Prevention is often our best path to fighting off viral infections. Quality water in the developing world would go a long way toward ameliorating the damage caused by rotavirus. They resemble a spoked wheel perforated by channels. Here in the figure for your textbook, you have the schematic composed of different viral proteins, VP1, you can see their name based upon their size, surrounding a double-stranded RNA genome with associated proteins. And then we see cutaway based upon electron cryo microscopy, and they have a spherical look to them. Its genome is segmented. 11 double-stranded RNAs. Each one encodes one protein except one which encodes two. Six of the proteins are structural, and 6 are non-structural.
What is that,
11.
11, okay.
So how can
Yes.
it be 6 and 2?
Yeah. Not all RNAs encode proteins.
Okay.
Oh, sorry, I'm sorry, wait a minute. That's the next virus. 11 double-stranded RNAs. All of them encode one protein, except one which encodes two.
Okay. So that's split.
I know it's been a long time since calculus. So 11 plus 1 is 12.
Okay.
6 are found in the final form, 6 are responsible for viral packaging. Yes, the next virus is not all encoded. So they're named, and I'm gonna have a brief discussion with you guys toward the end of this on how you want to handle this on the test. They're named VP in descending order of their size. So what's shown to you here is RNA bands and a gel electrophoresis experiment. And so these are the RNAs that encode the protein. So the largest RNA, which of course encodes the largest protein, is VP1, and so forth and so on. And they have different functions. So RNA polymerization is a complicated thing, so it stands to reason that the large one would be an RNA polymerase. And then of course, having nothing be simple, we can't have it be VP1 through 11, but rather there's VP1 through 7 based upon descending size, along with non-structural proteins. So the VPs are found in the final virion, and the non-structurals are not, and so they're named separately. They infect the villi. For example, in the gastroenteric system, so the villi in the small intestine would be where rotavirus goes. So these cells with a lot of surface area, ruffles on their apical membrane. What's the opposite of basolateral? I actually remembered apical. It's written right there. Upon the apical side, they're going to infect the cells. Here is an interesting example, at least to me interesting. And by the way, VP4 is the one that encodes 2. It's made as a polyprotein that's then cleaved hydrolytically into VP5 and 8. So this was originally named VP4 because there's an RNA, but really it's VP5 and 8 with the asterisk to denote that it's a proteolytic product. Attachment and entry varies from any virus we've seen thus far. So attachment can go through a cell surface protein and endocytosis, but interestingly, it's capable of direct penetration. So not receptor mediated, no real binding event in any way we've thought of. Here's something remarkable. How does a virus get inside a cell without binding to something on the surface? It does have a phospholipase. Yes, that would be one way. You have phospholipase on the surface that eats away at the cell membrane, but here we have depicted direct entry. It's via, there are actually proteins that are capable, they're called cell-penetrating proteins. Interestingly, I've just founded a biotech company based upon this. It's one of our good projects. That the proteins are capable of existing in both the aqueous milieu of the exterior of the cell and the hydrophobic core of the membrane. What you have is proteins that are at that balance between hydrophobicity and philicity, perhaps a structural conformational change as they go through, but there are proteins that can penetrate bilayers, and it's because they have an amphipathic quality to them, both charged and hydrophobic. And then through. So if we have endocytosis, that's where our phospholipase comes in to get out, because we're receptor mediated, so that would tend to limit contact between the lipid and the coat protein, but it doesn't necessarily have to be the case. Either way matters not. If it escapes the endosome, it's there in the cytoplasm.
Direct penetration is not receptor.
Yes, that's correct.
Do both of those things happen?
Yes. I can imagine, so I'm sure it varies depending on the reovirus, the affinity of the surface of the ligand and the receptor would matter. If we have a high affinity, we tend to go through there. If we have a lower affinity, we tend to see this being a rare event, but viruses make their bank off of rare events. So it being a double-stranded RNA, we can either have the plus strand, and this I believe is labeled inappropriately because the plus strand would be the messenger RNA, and then we could have in the cytoplasm, actually this virus never goes into the nucleus, we have off of the mRNA, we can have translation of all of the genome, and then the translation, the early translation from the messenger RNA results in the viral proteins that can synthesize RNA so that we then get formation of the polymerization of the RNA. At the same time, the other structural proteins are being made. They form a zone of inclusion within the cell, so this is not the nucleus. This is just an area in the cytoplasm where viral proteins start binding to one another because of their affinities. And then we have the viral assembly occur coinciding with the other strand of the RNA, such that some of the plus strand goes to mRNA, some of it goes to template for the minus strand so that we can get double-stranded RNA with structural proteins around it. And then we see escape of the viroplast. And then, there are other glycoproteins that have to go to the endoplasmic reticulum because that's where glycosylation occurs. And then assembly of the protovirus, or the precapsid, the protovirus with the glycoprotein in the endoplasmic reticulum. We can see what amounts to endocytosis in the endoplasmic reticulum, followed by another escape. So that the interior of the endoplasmic reticulum is the equivalent to outside of the cell, so transition outside the cell it occurs by escape and then transport of the vesicles to the cell surface.
What's those?
Sorry? That's transport to the cell surface. Sorry, I was pointing here, but it should be transport to the cell surface. Note I'm condensing the chapters to a couple of slides. So we have attachment not necessarily the case, we can have direct entry, which you can say is an attachment. Escape, translation, genome replication, assembly, which is both a soluble event of assembly of viral structural proteins and collection of the glycoproteins on the way out, which goes via the endoplasmic reticulum. It's the only way you can pick up the glycoproteins. And then the exit is a little bit different in that exit from the cell occurs at the cell membrane, but it's escaped from the vesicle in the endoplasmic reticulum that traffic within vesicles from the endoplasmic reticulum budding off to the Golgi body to vesicle transport. And out, it's already out when it's here. It's a naked virion.
Can it cross like all the membranes equally well, like since it can penetrate into the cell without any need for endocytosis? Does it really need any vesicles to get out of the other membrane organelles?
So this is I don't know. The phospholipase activity would have been reconstituted here because you've assembled the phospholipase into the capsid, so it could escape via phospholipolysis here. Here, that would be a less functional / non-functional virus because it would not yet have the glycoproteins, which are the things that are protruding for ligand attachment. So my guess is that it's only that this fusion with the endoplasmic reticular membrane is necessary, but it's necessary to pick up the glycoproteins because those are not synthesized in the cytoplasm. Glycosylation occurs at this. This is what you're seeing right here is the translation of VP7, which is the.
So it leaves the cell without actually destroying it?
Yes, that would be correct. Yes, it can leave the cell without actually destroying it, that's correct. But the disease is not based upon cell death but rather the stress this puts on, which causes osmotic changes between the villi and the exterior, so you lose water. Which is one reason why it's not a terrible health risk. If you pick up a rotavirus infection, you just stay hydrated. I have to ask my wife. I should have. She was at Journal Club last night, so I didn't get to talk about rotaviruses. I doubt she ever sees a patient with rotavirus infection. She's a gastroenterologist. So, like down her ass. Okay, well, let me back up. I guess what I want you to have coming out of this is that the idea, there's some new vocabulary here, the viroplasm, cell direct entry. So you should also be sensing some themes. That's it for chapter 13. I have rhabdoviruses in my head, sorry, that's what I looked at this morning. Coronaviruses, chapter 14. Plus strand, single-stranded RNA, class 4. Icosahedral geometry. Infectious largely in mammals and birds. Classic examples: Hep A, polio, some cold viruses, and then you can forget foot and mouth. Viruses with immense human health concerns and livestock / agricultural / economic ones. We have single-stranded RNA and a capsid, so we're naked here. A word about polio, I don't know, we'll come back to it in the course. So polio frequently causes less serious infections. It becomes serious, it becomes viremic or causes viremia or virus in the blood, and then gets to the central nervous system. When it gets to the central nervous system, we're looking at meningitis, encephalitis, and poliomyelitis. What does itis mean? So a meningitis would be a inflammation. You guys know what, and what is a meninges?
Lining of the brain.
Yeah. So an encephalitis would be an inflammation of the brain, and poliomyelitis would be an inflammation of the muscles caused by polio. And of course, this is the classic outcome of polio, is that we have paralysis, and you look at those terrible pictures in the book, and then respiratory distress caused by, respiratory failures caused by paralysis in muscles helping you to breathe. Like most viruses, no effective treatment, so vaccination against polio led to practical eradication. As your book notes, there's still occasional outbreaks of polio among the unvaccinated. say about the rhinoviruses and got my water. And foot and mouth. For the icosahedral, 60 individual subunits in the virion. The topological distinction of the icosahedron is these deep channels within the virus itself seen during structural electron cryomicroscopy of the virus. So that's what makes a coronavirus look like a coronavirus. Genome is 7 to 8 kilobases, so we're looking at very small things, not the most directly comparable to the parvoviruses. It's single-stranded RNA. 5' linked. Viral protein. G stands for genome. And then within the single-stranded RNA, there's a 5' untranslated hairpins that form ribosomal entry sites. So this is a plus strand single-stranded RNA, so upon infection, we have functional messenger RNA. And so here you have the protein, the untranslated region, they're showing you the hairpin structures. So this is these are Watson-Crick base pairing with areas where there's not base pairing, and these form binding sites for RNA polymerase to come in and translate the genome. So in our coronavirus, we have receptor-mediated endocytosis, escape from the endosome, whereupon we have removal of the 5' protein. We have functional messenger RNA so that translation can immediately occur. What we have is the equivalent of a polycistronic mRNA. So there are ribosomal binding sites, translation of different polypeptides, stop signal. So this is not a hydrolysis of a polyprotein, but rather translation of one protein followed by the next. Whereupon we have both structural proteins and proteins that will synthesize the minus strand of the RNA from which the additional plus strands can be synthesized. So in order to synthesize progeny genomes, we have to go through the synthesis of the minus RNA strand. So this is an RNA-dependent RNA polymerase. That's why it's extra nuclear. It has to be a virally encoded protein. This is not something eukaryotes do. When we have that minus strand, that's good as template for making the plus strand. So note that the plus strand can either be from the original infection, we get translation, or from progeny, we can get additional translations, which can also go back and form a template. Where we get progeny genome, production of enough proteins to assemble into the pentamer, procapsid, load the genome, additional maturation, followed by lysis of the cell and exit. One of the threats in the polio virus is that we can have recombination. So you can have viruses with different copies of the single-strand RNA that have different characteristics, different antigenic determinants. Viruses are very bad at copying themselves, so we see frequent induction mutations. You can have a recombinant coronavirus, polio being our prime example, where we have different antigens, and a recombinant virus may be resistant to future vaccines, so of serious concern. And the coronaviruses were the first ones synthesized in the lab in a cell-free environment, where if you take, give it cDNA, it can, in a cell lysate, it's an extract of a cell, given the genetic information, encode its own functional infectious viri, so they don't need the subcellular compartmentation, they just need the host proteins that they need. It's because it's a plus-stranded RNA virus. All right, I'm flying through it because I really want to get to stuff that interests me and interests you more. If you want to stop at this point and say, that's sort of the pace I want to cover the chapters at, you can go ahead and look at the details. I'm just going to try this for a while and see. I want to stop and ask, what the hell do you guys want to be tested on? So I'm starting to think about midterm 2. And you've now read, what have we read? 5 chapters. Each is, this is a virus, this is the geometry, this is the genome, this is the particular details of entry, attachment entry, transcription, translation, replication, assembly, and exit. I'm serious, what do you want? Do you want me to have multiple choice, match the viral protein with the virus?
Yeah. Yes.
Really?
Yes.
Golly. No, I'm going to ask you harder things as I think of them, but my instinct is to say, because I think you can tell, I don't really care that I know that there are 11 RNAs. See, I've already forgotten it. Whatever comes before coronaviruses. Reoviruses. Wrong.
Rhoda.
See, you didn't memorize it. So you guys really want that? So I guess.
Or mix the virus to the
So so those who are opposing the popular movement for minutia on the test are shaking their heads very, like they're at an auction bidding, you know, like. I'm getting some no's. I think what I'll do is put together a survey for you, so I can maybe let you know in a week or so. And again, it's not gonna be. There'll be some minutia. There'll be some big things.
Paper stuff.
Less paper stuff. Well, see, that's the part that interests me. How many of you read the paper for today? Yeah, I made an attempt. That's all. Yeah. Oh, okay. Yeah, it depends on how you define attempt, right?
Yeah. See the first page?
Choosing to have more than, what do you mean? Oh, like answer 3 of the following 5 questions?
No, instead of you being like, you either say yes or no, there is more than one thing.
Like, make a survey of sample questions.
Or like, kind of questions. Like, do you want these questions, or these, or these, or these?
Well, so it's going to be more of a weighting for you, right? So I'm not going to give you a test that's all minutia. If you look at what's available online for virology classes, it's seriously like,
I've looked at all.
you know, what's the phospholipase from rotavirus called? As I was talking to somebody before class, I've asked several physician friends of mine what they remember from virology, and uniformly the answer has been nothing. None of them are virologists or infectious disease specialists, but I would like it if 3 years from now you remembered something, and if that was learning a little how to read a paper, so much the better. Because I bet you're not going to remember whether rotavirus is a reovirus or a coronavirus. All right, any other feedback? Yes, ma'am.
Can you ask less about figures and maybe more about the figures of data?
I can, but I'm not going to. So you're seniors in biology. I think most / all of you have never seriously made an attempt at reading primary literature.
Never.
a good chunk of you want to be doctors, a good chunk of you want to go to graduate school. When you look at a paper like today's, it should, the first, I don't know, 100,000 times you look at a paper, it's like trying to listen to what dolphins are telling you. So getting into the literature and getting an example, today's is a good one, of where it is all this information comes from, to me is far more valuable than the actual information. If you become an infectious disease physician, fine. I will have served you poorly by not making you memorize the various flavors of single-stranded and double-stranded RNAs for the first many times. Those of you going to med school will see this. By the way, my buddy I was talking to about this, I said, what does he remember from virology? His answer wasn't nothing. It was same picture, different name. So anyway, I'll just take a feel for it. Less figure. So, from my opinion is 9% of your test was a figure. There's 9 points out of 100. That's not unreasonable, given that we're going to talk about more than one paper in this chunk of the course, and that I have an obvious disdain for the material we've talked about thus far. My tendency would be to weight it more toward the papers. I get that the vast majority of you are saying less, so maybe we'll keep it about the same. Let me take a survey and see what you think as a group when you're cloaked by anonymity. Because the guy who goes, I want more papers, might find his tires slashed. I want more papers. And I drive BMW right up here. I wonder how often guys in BMWs get their tires slashed because somebody tells them. Like Mercedes out there, yeah, that was. Mine's a really old Honda. Still doesn't eliminate it. Okay, so this was to be a cool paper. In that what we have is an example of how it is that a functional example of a part of the human genome being viral in origin, but yet having attained a significantly important function in our molecular biology. And so we start, as always, with the title, Human-Specific Endogenous Retroviral Insert, which will be our first little bit of vocabulary. hsERVs serves as an enhancer for schizophrenia-linked gene PROD-H. So that's, as a scientist, I hate viruses. Well, no, I shouldn't say that. I'm supposed to be enthusiastic for the subject. Viruses aren't my favorite thing. But that's a pretty provocative title. Viruses, schizophrenia? Is schizophrenia an infectious disease? Well, the answer is no, but that's kind of neat. So I'll just human-specific endogenous retroviral insert. Now, of course, we haven't talked about retroviruses yet, but those are the ones where RNA revert to DNA in order to be copied back into RNA for packaging, capable of insertion into the chromosome, which is in fact what happened here. So you read nothing else in a paper, read the abstract. So let's look at what they're doing here. They did a whole genome analysis of enhancers, so this is the bioinformatics approach, for these sorts of retroviral inserts and the previous information on what the sequences of these things look like. So they crunched the human genome. They're identifying something that's an enhancer element. Let's review what an enhancer element is. So it's going to increase transcription of prod, or there's one neurogene prod H. Proline dehydrogenase. So, what does proline dehydrogenase do? Dehydrogenates proline. What does that sound like that does?
Take the hydrogen off the proline.
Yeah, yes. Very good. You're all gonna do very well when you use the biochemistry. Yeah, I did already. But why would you dehydrogenate proline?
You can make it into something else.
So that's known as, well, it could be the other one, but, sorry, oops. Proline catabolism. You have a protein, it's outlived its usefulness, it's lost its structure, it's denatured. You have metabolic pathways that recapture that energy. So pathways that break down things are called catabolic, as opposed to the anabolic steroids I pump into my body before I lift. Anabolism is the synthesis, so anabolic steroids cue synthesis of things. I wonder if they are, I do probably catabolic steroids, but nobody goes, I need these muscles to be smaller.
You're probably losing weight or something.
That's known as amphetamines. Different pathway. So we're breaking down proline into something. So this could be, you've eaten too much, you've eaten some protein, you don't need the proline for protein synthesis, you have enough, you can catabolize it into something else. It happens to be that what you get out of it is, there's an intermediate here I'm not talking about, but you get glutamates. So you get another amino acid, which can eventually be synthesized into GABA. So that's not what not prod H. But proline dehydrogenase dehydrogenates proline into a product that can be used to synthesize glutamate, which can eventually be used to synthesize something called GABA. I bring it up because these two are neurotransmitters. So for example, the response to adrenaline goes through a GABAergic receptor, or can go through GABA. There are other proline dehydrogenases. This is the one, so it's not exclusive to this, but prod H is a proline dehydrogenase. So we've got an enhancer, we'll see in a second, it's near proline dehydrogenase. And they're discovering that it acts as tissue-specific enhancer required for proper CNS function. So prod H, as they note, is found in schizophrenic patients to occasionally be altered. So mutants, there's some non-coding mutants, so it's not necessarily the final protein. But people with schizophrenia have altered function of expression of prod H or an altered proline dehydrogenase, other neurological disorders as well. Woof, woof, woof. So you can woof, woof, woof your way through some stuff. We showed, that's always very important to stop woofing when you see this, that this element is an enhanced, the element, and there's another CPG island, we'll talk about that in a minute. They synergistically, they work together to activate the promoter of prod H. It's regulated by methylation, talk about that in a minute. Under methylated, it can upregulate in the hippocampus. I don't know about anything above bacterial cell level, but this is a pretty important part of the brain. Go talk to Dr. Reese or someone else if you want to know exactly what the hippocampus does. It's part of your brain where you think about hippopotami. So there's another transcription factor, SOX2, which we'll find is neural specific, preferentially expressed in the hippocampus. We propose that the interaction of this viral origin DNA and this human enzyme may have contributed to the evolution of the central nervous system of humans. That's pretty cool. Let's see if we believe it. Right? Our brains are structured the way they are because we were infected with a virus. Might be a good plot for science fiction. Oh yeah, I guess 12 monkeys is already good. So what they mention the results, but there's no figure for, is that within the human genome there are 103 HS or ERV sequences. They don't know that they're actually hsERV sequences, they're just by whatever screen they set up, we have information on what the retrovirus looked like. And so these elements should look like the sequence X. There are 133 potential sequence X's in the human genome. Before I get to that, it was, sorry, I already got to it. Let me pretend like I haven't. Chimpanzee and gorilla genomes were, this isn't in the paper, this is were bombarded, this is in one of the references mentioned in the introduction to the paper, bombarded with retroviruses, retroviral sequences 3 to 4 million years ago. And so the theory is that some of these sequences, the vestiges are sort of non-productive latent infections of the virus, by the virus in the human genome. So it's sort of the garbage that's left over after viruses have very poorly infected the genome. Little bits and pieces break off. 133 potential HRVs in the human genome. They looked at the entire genome sequence, and by their definition, which is less than 5 kilobases, there are 6 ERVs near known genes. I don't know if you've ever talked about the junk DNA in the human chromosome or human genome. There's a bunch of DNA that doesn't code for any protein. Why is it there? One of the theories is for defense against these sorts of things. If you have a bunch of DNA that's not sequenced, you're giving viruses a target that when they integrate in the chromosome don't disrupt function. So look, it looks like it served pretty well. 133 in leftover latent infections, 6 of them even near something that seems to matter, at least to us. Of those, hang on, 3 is the number I'm looking for. Let's get to figure 1, and we'll come back to what 3 are important. So here's another method not mentioned in your text. So they're going to do luciferase assays. Anybody know what luciferase is?
Enzymes that are responsible for making certain organisms glow.
Yes, so firefly butts have luciferase in them. So if you give them their substrate, they glow. So they produce light. You can measure evolution of light. So the assay is you have a plasmid that either has the upstream region of the gene that's near the hsERV, or you have a plasmid that has the upstream region near the hsERV plus the hsERV. So they're calling this LTR minus LTR plus. I couldn't quite figure out, this is a very heavily Russian paper. I don't know if that's some transliteration from Cyrillic or not, or Cyrillic alphabet to English. There's one guy in there from Boston and about 15 Russian letters. You put that immediately upstream of luciferase in a plasmid, so that what you get is the regulatory, the promoter and enhancer elements nearby upstream of the gene. So you can turn on luciferase instead of, in this case, prod-H or the other genes that it's near. And rather than having to measure proline dehydrogenase activity, you measure light evolved. And so you have either the promoter and nearby enhancer regions, like the minus 35 and minus 10 sequences that we use in general biology, or all that, plus the element. And then you transfect this into cells. So transfection is like infection with the virus, but instead of a virus, you give it a plasmid, the circular DNA. Mammalian cells will, under certain conditions, take up foreign DNA, and this is it'll just exist as an extra chromosomal piece of DNA for a while. If you have a functional promoter gene, you'll get gene expression. And so, here you go. So they're looking at normalized promoter activity, so the proline dehydrogenase, LTR plus, LTR minus, and then the same prod H sequence from chimpanzees. So we're getting the human specific, so that's why they're bringing in chimpanzee. And so they looked at proline dehydrogenase in several different cell lines. TERA1 is the one that's of interest, but for example, I didn't look up these other ones. HepG2 is an immortal cell line from somebody who died of liver cancer in the 1960s. I believe it was a 17 or 18-year-old boy, teenager, I guess. And his cells have been kept alive ever since. So the immortal cancer cell lines make good lab cell lines because they grow uncontrollably as opposed to normal cell lines which die. So they're not working in healthy cells, they're working in cancer-ridden cells, and then the TERA I made a note and I can't find it. TERA are not neurons, but happen to have the activity. So they're just taking a guess here. It turns out there's a retroactively interesting reason, but I lost what it is. But they're comparing prod H activity or promoter activity, so the promoter ability to turn on luciferase relative to the presence of this sequence. So what we see here is a lot more when you have the LTR than when you have the LTR minus, and then if you had the chimpanzee. So these are human cells preferentially expressing the enhancer. So this is showing you that you have enhancer activity. And then these other cells, you see some activity, but doesn't look like there's much difference. So the enhancer isn't really doing much of anything there. HepG2 cells even less. The other genes that have these hsERVs are shown in the supplemental figures, which I didn't look. So there were 3 candidates that had any effect at all. Prod H was the one that had the very pronounced effect. Oh, there it is. So looking at these were other. So these were the other genes. So SOC-S4, Prod H, KII 1919. These were the 3 of the 6 that had any increase in expression at all in the TERA. But what you see here is that then you're getting luciferase activity. If you look at the messenger RNA, they're not tissue specific. This one is the one that's tissue specific. So they're getting an upregulation in all the cell lines at the mRNA level. So this is a translational difference. When you see an increase in luciferase activity, it could be because you're making a little bit of RNA that's being translated a lot, you're making a lot of RNA that's been translating little, you translate a little, or you're making a lot of mRNA that's been translating a lot. In this case, we see mRNA produced in response. So this is actually a better measure of promoter activity because there's some translational regulation going on elsewhere. So take-home point here is the hsERV upregulates luciferase in those constructs in a tissue-specific manner. TERA is a tissue derived from lung cancer cells, and that at the mRNA level, the TERA hsERV is the one that matters. So within the Prod H gene, overlapping a little bit with exon 2, so remember exons and introns have to be spliced. Getting rid of the introns and exons is a CPG island. This is a new one to me as well as I read the paper. So CPG island is an area, it's about a 300 to 3,000 base pair region, with, as you would guess, heavy CG content. The general cutoff is 60% CG as opposed to 18. Now, the P there is for phosphodiester that means they're linked by a phosphodiester bond. That's to differentiate from a CG base pairing on different strands. So CG are next to one another on the same strand. It's found in, as a regulatory element in about 40% of mammalian genes. So downstream from the promoter within the coding region is a CPG island in prod H. By the way, one other thing I skipped over is authors use the word etiology. Does anybody know what that means? As a biologist, you ought to know what etiological or etiology means. The set of causes. Just as a matter of your vocabulary as upstanding citizens in the 21st century. So, and they use it here, or sorry, in the previous figure to talk about the prod H, hsERV in Tera cells was the only one to have a 7 to 10-fold change when accounting for the cell lines of different etiologies. So the HepG2 has a different etiology than the Tera1 cells. So, made a bunch of different luciferase constructs that either had that, this is the equivalent of the plasmids from the first two, or containing just the CPG islands, they have a little bit of the coding sequence here, versus both hsERV and the CPG island. And what you see is, here's their response due to the hsERV, here's their response due to the CPG island. It's a little bit more than if nothing were there. But when you have them both together, you get a synergistic increase in the amount of prod H promoter activity. So this is evidence that the two elements, the CPG island and the retroviral insertion, are acting together. Okay, so they obtained a brain from a single donor. They have absolutely, I look back, I'm like, I gotta see, where did this come from? And they don't mention it. So it's Russia, right? I have a little note margin, insert Ukraine joke here, but I won't. No, I imagine somebody probably died in a motorcycle crash in Moscow, because all the universities were in Moscow and they were able to get a donor from a recently deceased person. So if you check your organ donor on your driver's license, you could one day wind up in a paper tube. But why they only look at one brain, I presume, because human brains that are recently deceased tend to be hard to come by. And so they're looking at RNA from a brain. That's why it's really hard to come by. DNA is very, very stable. RNA isn't. They got a human brain, and then took tissues from the various anatomical regions, including our hippocampus, right there where he's thinking about hippos. Maybe that's why he had a motorcycle crash. Imagining those hippos like from Fantasia dancing. No attention span whatsoever. And then we're looking at hang on a second. B. I'm sorry, I tend to go AB, and I'm going AB. Looking at methylation of the, I'm sorry, they are looking at DNA here. Looking at DNA methylation of the CPG island. So one bit of an aside is, so, let me start here. you have methylation enzymes that will methylate both CPG and hsERV sequences. You can do a DNA sequencing method that tells you whether something has been methylated or not. They did some in vitro studies where they methylated the DNA of those constructs with a purified enzyme, and both in the in vitro studies, methylation, CPG. When the CPG sequence was methylated, they saw less activity in the Prod H promoter. When the hsERV was methylated, they saw a downshift, a decrease in activity. They then looked trying to remember what this thing is about. An expression of Prod H in these various sections they took. So they're doing QT, QRT, PCR. The quantitative reverse transcription PCR. So this is a way of measuring how much messenger RNA is in a sample. And not surprisingly, given that we've already read the abstract, we learned that the greatest amount of RNA is expressed in the hippocampus. And then there's some other places that they deem are important. But hippocampus, most of the Prod H RNA. And then go to C. And note that methylation is, this is the methylation pattern of those DNA sequences in hippocampus and either the frontal lobe, so this is their lower sample. We see methylation of the hsERV sequence very heavily, and then not as much activity, and then methylation to CPG less so, but a function there. So that hsERV is that the take-home point. hsERV is preferentially active in the hippocampus. So remember, they act synergistically. Just one being on isn't enough. Both of them are on in the hippocampus. Unless we see. I'm going to skip over this. This is an additional study of methylation. But that we then look at brain tissues from the single human donor and look at Prod H transcription, the hippocampus, the prefrontal cortex. So these were the things that were deemed relatively high there in humans versus a chimpanzee brain, which is presumably easier to get. We see the human specificity. So the chimpanzees have these element sequences in them, but they're not active in the chimpanzee. Forgive me, because last night I was looking at a photocopy. I didn't have the fluorescent in there, so I'm going to have to figure this out as I go along. So they're looking at primary cultures of the hippocampus. So the hippocampus is an anatomical structure in the brain. It has different kinds of cells in it. They transfected with two different plasmids, one of which was green fluorescent protein off of a strong constitutive promoter. So it's always expressed. So if it goes in to a cell, it's green. So this cell is delineating so this is a marker for the cell has been transfected. They have a mix of two plasmids, the other of which is Prod H promoter in front of something called DS red. It's red. So when you have a cell transfected with DS red, the DS red is under the neuron. So we have a neuron in the hippocampus transfected with the Prod H under its hsERV. We see more expression in the neurons, you put them together, red and green, I think yellow, that's right?
Yeah.
So, good, yellow. So we see in the neuron expression of prod H. Here, we see the cell's defined, it's been transfected, but we see no prod H expression. It's in an astrocyte, a different kind of cell present in the hippocampus. Same is true of the microglia, we get no expression. So we have neural specific expression of the reporter construct. So there's something in those neural cells of the hippocampus that allows for expression of proline dehydrogenase, again, via these two elements, the hsERV and the CPG island. And this is also projecting very poorly. It looks awesome on the slide, so I'll leave that to you to download. So what's an enhancer do? It turns out, it turns out gene expression. How?
Binds to the enhancer sequence or promoter.
The enhancer binds to what?
Could be enhancer sequence or promoter.
A transcription factor. So the enhancers are I'm sorry, I was just trying to introduce the next topic. I know it was a dumb question. Transcription factor binding sites. They looked at what transcription factors are known to bind within the hsERV sequence, and there were two of them that were candidates, SOX2 and NFKB1. Yes, 1. So these are transcription factors that are expressed in neural tissue. And the take-home point is, so when you co-transfect TERA1 cells with your prod H construct, you get this amount of activity, so that's what we saw before. But when you transfect with a plasmid-contained SOX2, so you're overexpressing. This is not, TERA cells are not neurons, they're derived from lungs. But when you give it both the prod H promoter, hsERV enhancer that's neuron specific, and the thing you think might bind to it that's expressed in neurons, even though it's not a neuron, you turn it on because you're building all the parts necessary to turn on that gene. Of course, we're not turning on prod, we're turning on luciferase because we can measure. We see SV40, this is a viral promoter, simian virus 40 promoter. This is one standard used in tissue culture to drive expression of whatever protein of interest you're in. So this is a constitutively active promoter. So we see by comparison, if you don't have the hsERV sequence, you get bupkis. Well, there's not quite bupkis there, but basically the same as if you have no promoter at all. If you have both the enhancer, promoter, enhancer, and transcription factor overexpressed, you get even more expression. So that's very, very strong evidence. This is the other one. They don't see much of a shift. That you get some. Sorry, this is another. I was thinking this was in FKB1. SOX2. This is the other cell. It's not tissue specific. We see less of a. I'm sorry. This is another cell line that did not have the same mRNA profile. But again, we get neural promoter with the SOX and the hsERV. We see more, not as strong of an effect. There's something going on at the mRNA level. They also do, this is supporting activity and electrophoretic mobility shift assay. This is a binding assay that's qualitative, where you mix radio-labeled DNA with a protein, and then you run it in gel electrophoresis on a native gel, so that the protein doesn't unfold. So you have DNA, you can see this, it's labeled, and DNA plus anything that binds DNA. In this case, it's a transcription factor. You can do this with polymerase, for example. And if it binds, this is all there is to it. This is bigger than this, so it runs slower on the gel. And so when we have, and there are two SOX2 binding sites in the element. When we see some shift, or not much shift, they claim there's one here, with either site as separate sites, and then a control site that has a shift. So this, you're mixing an extract. This is an extract where we have a transcription factor that binds to some other elements. That's, yeah, the experiment's working. Without the transcription of SOX2, we don't see any shifts of our DNA sites, except in the control. But with the other strain, so the other cell line and the neuron-specific site, we see a shift. So this is evidence that we're turning the gene on. This is direct evidence that it's SOX2 binding to that sequence of DNA. And then they change the binding sites. They use an altered DNA, so it's the equivalent of mutation. They lose the binding, so they don't see this shift. They then purify SOX2. They're not using an assay, an extract, and sure enough, it shifts where it doesn't shift if there's a mutation. So to sum up, they have identified the viral site, showed that it turns on gene expression, synergistically acts with another element that's not viral in origin, that it's neural specific, such that we have proline dehydrogenase expressed in a large amount relative to the viral sequence not being there in brain tissue. Does that matter? So they don't know. So their evidence doesn't tell us anything. Remember the title? Nothing they've talked about, that was all their data, except for their supplemental figures. The supplemental figures are all the experiments that didn't work. These six, remember the 6 elements near genes, they show you the 3 that didn't have any effect in the supplemental figure. Schizophrenia linked gene, they haven't said anything about schizophrenia. That's because they didn't actually do anything about schizophrenia. I'm sorry, I don't have the figure here because it's in the discussion. But they go into the literature, so what is known? A deletion in the genomic locus in chromosome 22q11, which contains the PROD-H gene, has been implicated in DeGeorge syndrome and associated with several other neurological disorders, including schizophrenia. So very well established. PROD-H alterations have neuropsychological effects and are implicated in the disease. So they're working with the sequence that's been shown to affect neurological disorders. And they hypothesized, so here's what made it important. By the way, PNAS is a huge journal. If you're in the Proceedings of the National Academy of Sciences, at least USA, if you're in the Proceedings of the National Academy of Sciences of Somalia, it's not as big a deal. Actually getting it published might be. But the PNAS is one of the leading journals, so this is an important study, at least it's deemed to be by the people who reviewed it. We hypothesized that at some point in human evolution, the hsERV insertion may have influenced PROD-H transcriptional activity significantly and increased its expression in the CNS. And SOX2, that transcription factor, is expressed preferentially in hippocampus, which is known to be one of the brain structures most effective in schizophrenia. Our findings suggest the potential role of the hsERV near PROD-H in CNS function. Discovery of this CNS-specific PROD-H enhancer encourages further studies to discover the molecular features affecting its functional activity in connection with neurological disorders. So this paper is a great example of something in virology actually matters. Something in virology, so a virus-derived DNA sequence having affected human evolution. Right? We need this viral element to our DNA in order to have a brain that functions properly. At least that's the hypothesis. You know, we need additional experimentation. Perhaps that may lead to eventual genetic therapy for people who are at risk for schizophrenia. If you can turn up their PROD-H expression, is that correct what's wrong with their brain? And who knows, probably not, but maybe. And that's sort of the halting nature of science, right? A little promise, something eventually will happen or breakthrough. So, encourage you to read the paper. The other reason I keep picking PNAS papers is not only are they very important, believe it or not, they're short. So if you didn't read the paper, please go read it. Next week's is posted, I'll put a news thing, it's a biotech paper, too and short.
You had a professor or a tutor for physics, and you had calc-based.
No, I had tree based, but he teaches calc based. I just don't know how much he would be available, because I know he just got a new job, but I can give you his number.
Yeah, that's great.
He's awesome. Like, he's he really is just like amazing.
Yeah, I just seen somebody that like explain this to me. So I dumbly took it.